Dear students, the topic of our lecture is Physiology of synapses and interneuronal connections. In this topic, we continue discussing the function of nerve cells and especially their ability to conduct impulses to the other cells in the central nervous system and to the other excitable tissues. In general, in the central nervous system, information is transmitted mainly in the form of nerve active potentials, which also are called as nerve impulses. And due to the connection which exists between neurons, this impulse can travel from one cell to another. And each impulse may be blocked in its transmission from one neuron to the next. Impulse may be changed from a single impulse into repetitive impulses, or impulse may be integrated with impulses from other neurons, and all these functions actually can be characterized as synaptic functions of neurons. Here in the slide, you actually see the scheme of neuron, the branches, dendrite, axons, and depending on the point of communication, we can discuss different kinds of synapses. So synapse, this is a specialized area of contact between two excitable cells. In this case, we are discussing the neurons. And transmission of information between these two neurons take place by different mechanisms. There are electrical and chemical synapses, and in this lecture, in more details, I will explain how they function, what are their specialties. So, first of all, let us point attention to classification of synapses. Synapses may be classified by their location. There are peripheral synapses. They may be neuromuscular or neurosecretory. This is because existing in human organism just three kinds of excitable tissues, which are the nerve tissue, the muscular tissue, and secretory cells. And because of that, in periphery, we can obtain just two kinds of connections of neuron to peripheral tissues, or to muscles, or to secretory cells. In the central nervous system, neurons may create contacts using different branches and different parts of cells, and so there are central neuronal synapses, which are exoaxonic, exodendritic, exosomatic, or dendritic. Synapses also may be classified by their functioning, because in the postsynaptic membrane, the result of conduction impulse can bring different consequences. This may be excitation or inhibition. And because of that, we can classify exciting synapses or inhibitory synapses by their function. By mechanism of impulse conduction, there are electrical synapses, chemical synapses, and combined electrochemical synapses. And most of the synapses used for signal transmission in the central nervous system of the human organism are chemical synapses. Especially they are spread in peripheral connections, and also they are very numerous in the central nervous system. In this kind of synapses, the first neuron secretes at its nerve ending in the synapse the chemical substance called a neurotransmitter, or sometimes called as mediator substance. And this neurotransmitter then acts on the receptor in the postsynaptic membrane. These receptors are proteins according to their nature, and so the neurotransmitter binds to receptor and can provide or excitation of postsynaptic cell or inhibition of postsynaptic cells, so the sensitivity of the postsynaptic cell may be modified as a result of this action. And nowadays in physiological research literature described more than 50 important neurotransmitters, and some of them are much more spread in the organism, like acetylcholine, norepinephrine, epinephrine, histamine, gamma aminobutyric acid or GABA, glycine, serotonin, and glutamate. In electrical synapses, the cytoplasm of adjacent cells are directly connected by clusters of ion channels called gap junction that allow free movement of ions from the interior of one cell to the interior of the next cell. Actually, in the picture, you may see in the case B the electrical synapse, and you can see how is different structure of electrical synapse, and it's understandable that this kind of connection between neurons provides much faster conduction of information between these two cells. Although most synapses in the brain are chemical, electrical and chemical synapses may coexist and even interact in the central nervous system. And the bidirectional transmission of electrical synapses permits them to help coordinate the activities of large groups of interconnected neurons, and this gives faster and more special conditions for processing information by neurons. Actually, as you already understand, nerve impulses which are spread between nerve cells exist actually for exchange by biological information. Electrical synapses are useful in detecting the coincidence of simultaneous subthreshold depolarizations within a group of interconnected neurons, and this function enables increased neuronal sensitivity. It promotes synchronous firing of group of interconnected neurons, and this helps for more intensive and better processing of information in the brain. And the specialty of the chemical synapses, as for their functioning, may be characterized as one-way conduction. You actually could see in the slide the principal structure of the chemical synapse, and you should know that chemical synapses have one exceedingly important characteristic that makes them highly desirable for transmitting nervous system signals. This characteristic is that they always transmit the signals in one direction, that is from the neuron that secretes the neurotransmitter, called the presynaptic neuron, to the neuron on which the transmitter acts, called the postsynaptic neuron. This phenomenon is the principle of one-way conduction at chemical synapses, and it is different from conduction through electrical synapses, which often transmit signals in either direction. A one-way conduction mechanism allows signals to be directed towards specific goals. In this specific transmission of signals to discrete and highly focused areas, both within the nervous system, at the terminals of the peripheral nerves, that allows the nervous system to perform its myriad functions of sensation, motor control, memory, and many other functions. And actually in the picture you can see the structure of actin chemical synapse and the important items of the conduction impulse, which we will discuss a little bit later. In classical example, we discuss function of chemical synapse based on principle of conduction excitation by cholinergic synapse. First of all, necessary to discuss function of presynaptic terminals. You can see on the slide the electronic microphotograph, which shows to you the organelles present in the presynaptic terminal, and the photograph shows that terminal has varied anatomical forms, but most of them resemble small round or oval knobs, and therefore are sometimes called terminal knobs or synaptic knobs, buttons, and feet, or synaptic knobs. And presynaptic terminal is separated from the postsynaptic neuronal soma by a synaptic cleft, usually 200 to 300 angstroms wide. The terminal has two internal structures important to the excitatory or inhibitory function of the synapse: the transmitter vesicles and the mitochondria. They are actually presented in the picture. The transmitter vesicles contain the neurotransmitter that, when realized into synaptic cleft, excites or inhibits postsynaptic neuron. It excites the postsynaptic neuron if the neuronal membrane contains excitatory receptors, and it inhibits the neuron if the membrane contains inhibitory receptors. The mitochondria provide adenosine triphosphate, or ATP, which in turn supplies the energy for synthesizing new transmitter substances. When an action potential spreads over a presynaptic terminal, depolarization of its membrane causes a small number of vesicles to empty into the cleft. The released transmitter, in turn, binds to a receptor on the postsynaptic neuronal membrane, causing an immediate change in its permeability characteristics and leading to excitation or inhibition of the postsynaptic neuron, depending on the neuronal receptor characteristic. There is considerable variation in the anatomic structure of synapses in various parts of the mammalian nervous system. The ends of the presynaptic fibers are generally enlarged to form terminal buttons or synaptic knobs. Endings are commonly located on dendritic synapses, which are small knobs projected from dendrites. In some instances, the terminal branches of the axon of the presynaptic neuron form a basket or net around the soma of the postsynaptic cell, and there is term basket cells, which you may remember from histology. For instance, in the cerebellum, you could reveal such kind of cells, or also in autonomic ganglia. In other locations, they intertwine with the dendrites of the postsynaptic cell. Climbing fibers of the cerebellum may be example of such kind of connection, or they end on the dendrites directly. For instance, apical dendrites of cortical pyramids, also which you may remember from histology, or on the axons, like exoaxonal endings. In the spinal cord, the presynaptic endings are closely applied to the soma and the proximal portions of the dendrites of the postsynaptic neuron. The number of synaptic knobs varies from 1 per postsynaptic cell. For instance, this is revealed in the midbrain, to a very large number. The number of synaptic knobs applied to a single spinal motor neuron has been calculated to be about 10,000, with 2,000 on the cell body and 8,000 on the dendrite, for example. Indeed, there are so many knobs that the neuron appears to be encrusted with them. And the portion of the soma membrane covered by any single synaptic knob is small, but the synaptic knobs are so numerous that in aggregate, the area covered by them all is often 40% of the total membrane area. In the cerebral cortex, it has been calculated that 98% of the synapses are on dendrites and only some 2% are on cell bodies. The transmitter release from the presynaptic terminal may be done due to action of calcium ions. This is important item. I also wish to note that you need point special attention to the mechanism of conduction excitation in chemical synapse, because this knowledge is essential for understanding pharmacology, mechanism of action of variable pharmacological substance originates from the physiological processes in synapse. So, the membrane of the presynaptic terminal is called the presynaptic membrane. It contains a large number of voltage-gated calcium channels, and when action potential depolarizes the presynaptic membrane, these calcium channels open and allow the large number of calcium ions to flow into the terminal. The quantity of neurotransmitter that is then released from the terminal into the synaptic cleft is directly related to the number of calcium ions that enter. The precise mechanism whereby the calcium ions cause this release is not known, but it is believed to be the following. The calcium ions, when they enter the presynaptic terminal, bind with special protein molecules on the inside surface of the presynaptic membrane called release sites. This binding in turn causes the release sites to open through the membrane, allow a few transmitter vesicles to release their transmitter into the cleft, after each single action potential. And for vesicles that store the neurotransmitter acetylcholine, between 2,000 and 10,000 molecules of acetylcholine are present in each vesicle. And there are enough vesicles in the presynaptic terminal to transmit from a few hundred to more than 10,000 action potentials.
A single brain cell or neuron. Here is an actual neuron, magnified 10,000 times. Neurons are the tiny building blocks of the brain. They do something remarkable which prompts all our thoughts. They fire an electrical impulse. Amazingly, we can now see one firing. This is the first time it's been shown on television. The electricity is bursting along the neuron at 400 kilometers an hour. Here we're seeing it in slow motion. Within a tiny fraction of a second, it's ready to fire again. Your brain has a staggering 100 billion of these neurons. Together, they could generate enough electricity to illuminate a light bulb. To make things more complicated still, the branches of each neuron are connected to thousands of other neurons. It's hard to grasp the sheer scale of all these connections. And there's more. Go deeper into this tangle, travel along a single neuron, and take a close look at the junction with its neighbor. Oddly, the neurons are not physically joined together at all. There's a tiny gap. To bridge this gap, the neurons release minute quantities of chemicals every time they fire. Chemical go-betweens that influence our thoughts.
The transmitter action on postsynaptic neuron has also specialties depending on the kinds of receptors, protein receptors which are present in the postsynaptic membrane. So the membrane of postsynaptic neuron contains large number of receptor proteins. The molecules of these receptors have two important components. First of all, a binding component that protrudes outward from the membrane into the synaptic cleft, where it binds the neurotransmitter coming from presynaptic terminal. At second, there is an intracellular component that passes all the way through the postsynaptic membrane to the interior of the postsynaptic neuron, and receptor activation controls the opening of ion channels in the postsynaptic cell. There are two ways of opening these postsynaptic cell channels. First of all, by gating ion channels directly and allowing passage of specific types of ions through the membrane. At second variant, by activating a second messenger that is not an ion channel, but instead is a molecule that protrudes into the cell cytoplasm and activates one or more substances inside the postsynaptic neuron. These second messengers increase or decrease specific cellular functions. Neurotransmitter receptors that directly gate ion channels are often called ionotropic receptors, whereas those that act through second messenger systems are called metabotropic receptors. The second messenger system in the postsynaptic neuron has its functional peculiarities. Many functions of the nerve system, for example, the process of memory, require prolonged changes in neurons for seconds to months after the initial transmitter substance is gone. The ion channels are not suitable for causing prolonged postsynaptic neuronal changes because these channels close within milliseconds after the transmitter substance is no longer present. However, in many cases, prolonged postsynaptic neuronal excitation or inhibition is achieved by activating a second messenger chemical system inside the postsynaptic neuronal cell. And then it is the second messenger that causes the prolonged effect. There are several types of second messenger system. One of most common types uses a group of proteins called G proteins. You actually can see in the picture the second messenger system which uses the G protein. And inactive G protein complex is free in the cytosol and consists of guanosine diphosphate plus 3 components. An alpha component, that is the activator portion of G protein, beta and gamma components that are attached to the alpha component. As long as the G protein complex is bound to GDP, it remains inactive. When the receptor is activated by a neurotransmitter following a nerve impulse, the receptor undergoes a conformational change, exposing a binding site for the G protein complex, which then binds to the portion of the receptor that protrudes into the interior of the cell. This process permits the alpha subunit to release GDP and simultaneously bind guanosine triphosphate while separating from the beta and gamma portions of the complex. The separated alpha GTP complex is then free to move within the cytoplasm of the cell and perform one or more of several functions depending on the specific characteristic of each type of neuron. Inactivation of the G protein occurs then the GTP bound to the alpha subunit is hydrolyzed to GDP, that is guanosine diphosphate. This action causes the alpha subunit to release from its target protein, thereby inactivated the second messenger system, and then to combine again with the beta and gamma subunits, returning the G protein complex to its inactive state. It is clear that activation of second messenger system within the neuron, whether of the G protein type or other types, is extremely important for changing the long-term response characteristics of different neuronal pathways. And now let us summarize the sequence of events about conduction of impulse by chemical synapse on the example of cholinergic synapse. So, the item first, the action potential travels down the motor neuron, depolarizes the presynaptic membrane, and in the picture, the red areas on the presynaptic membrane actually shows the action potential transduction along the nerve fiber. Item second, this depolarization opens voltage-gated calcium channels in the presynaptic membrane, resulting in calcium ions influx into the presynaptic terminal. Item 3. The rise in calcium causes synaptic vesicles to release their contents, in this case acetylcholine, and the amount of neurotransmitter release is directly related to the rise in cytosolic calcium. So the more calcium enters the presynaptic nerve ending, the more vesicles can open and release their neurotransmitter. And item 4, this is the time when acetylcholine binds to a nicotinic receptor located on the muscle membrane, so-called NM receptor. The NM receptor is non-selective monovalent cation channel, both for sodium and potassium traverse. And given that sodium has a much greater net force, depolarization occurs. And this depolarization is called an end plate potential, or you may meet abbreviation EPP. The magnitude of the end plate potential is directly related to the amount of acetylcholine released. Actually, the intense of effect depends on number of vesicles to be opened. The more acetylcholine revealed into synaptic cleft, the greater effect. And the resulting depolarization opens fast sodium channels on the muscle membrane, named sarcolemma, causing an action potential in the sarcolemma. Under normal circumstances, an action potential in the motor neuron releases enough acetylcholine to cause the EPP that is at least threshold for the action potential in the skeletal muscle cell. In other words, there is a one-to-one relationship between an action potential in the motor neuron and action potential in the skeletal muscle cell. The actions of acetylcholine are terminated by acetylcholine esterase. This is an enzyme located on the postsynaptic membrane that breaks down acetylcholine into choline and acetate. Choline is taken back into the presynaptic terminal, so there is process of reuptake of choline, hence providing substrate for the resynthesis of next portions of acetylcholine. The circulation of substances in the same synapse may take place till 20 times. And this saves energy and saves materials in synapses. Synapses between neurons may be characterized by certain specialties of the functioning, and in general, the synaptic potentials produced are excitatory or inhibitory, and they are produced by ligand-gated ion channels. In this kind of synapses revealed their location on the cell body and dendrites. The currents produced at these synapses travel along the dendritic and cell body membranes. The axon hillock, which is initial segment region, which has a high density of fast sodium channels, and this portion of cell helps to give origin for the action potential of the axon. The closer the synapse is to its region, the greater its influence in determining whether an action potential is generated. If the sum of all the inputs reaches threshold, and action potential is generated and conducted along the axon to the nerve terminal. On activation, some postsynaptic receptors cause excitation of postsynaptic neurons, and other may cause inhibition. So result of action varies depending on neurotransmitter and kinds of receptors to it. The importance of having an inhibitory and excitatory types of receptors is that this feature gives an additional dimension to nerve functions, allowing a restraint of nervous action and excitation. Excitatory postsynaptic potential, or EPSP, is excitatory if it increases the excitability of the postsynaptic neuron. That is, it is more likely to fire an action potential. It is primarily the result of increased sodium conductance. It is similar to the EPP found at the neuromuscular junction. If to characterize excitation process, opening of sodium channels to allow large numbers of positive electrical charges to flow to the interior of the postsynaptic cell, and this action raises the intracellular membrane potential in the positive direction up toward the threshold level for excitation. Depressed conduction through chloride or potassium channels decreases the diffusion of negatively charged chloride ions to the inside of the postsynaptic neuron. This action also decreases the diffusion of positively charged potassium ions to the outside. In either case, the effect is to make the internal membrane potential more positive than normal, which is excitatory. Various changes in the internal metabolism of the postsynaptic neuron to excite cell activity, or in some cases to increase the number of excitatory membrane receptors, or decrease the number of inhibitory membrane receptors. Inhibitory postsynaptic potential, or IPSP, is inhibitory if it decreases the excitability of the postsynaptic neuron. That is, it is less likely to fire an action potential. It is primarily the result of increased chlorine conductance. Let us describe the sequence of events when inhibition develops. First of all, opening of chloride ion channels through the postsynaptic neuronal membrane. This is the action which allows rapid diffusion of negatively charged chloride ions from outside the postsynaptic neuron to the inside, thereby carrying negative charges inward and increasing the negativity inside, which is inhibitory action. Increase in conductance of potassium ions out of the neuron also may take place. This action allows positive ions to diffuse to the exterior, which causes increased negativity inside the neuron, and this also produces an inhibitory effect. There is also effect of activation of receptor enzymes in case of inhibition. This process inhibits cellular metabolic functions and increases the number of inhibitory synaptic receptors, or decreases the number of excitatory receptors. During transmission of a neuronal signal from a presynaptic neuron to a postsynaptic neuron, a certain amount of time is consumed in the process of discharge of the transmitter substance by the presynaptic terminal. Also, some time required for diffusion of the transmitter to the postsynaptic neuronal membrane. The next event which takes time, this is action of the transmitter on the membrane receptors. Also, time is required for action of the receptor to increase the membrane permeability. And finally, invert diffusion of sodium to raise the excitatory postsynaptic potential to a high enough level to elicit an action potential also takes some period of time. Actually, these all events which I have listed before, this is the reason for so-called synaptic delay. Synaptic delay, this is the time which takes the process of conduction impulse from one cell to the other through the chemical synapse. In chemical synapse, this synaptic delay is much greater than in the electrical one. When an impulse reaches the presynaptic terminals, there is an interval of at least half a millisecond, which actually consists the synaptic delay, before a response is obtained in the postsynaptic neuron. The delay following maximal stimulation of the presynaptic neuron corresponds to the latency of the excitatory postsynaptic potential and is due to the time it takes for the synaptic mediator to be released and to act on the membrane of the postsynaptic cell. Because of it, conduction along a chain of neurons is slower if there are many synapses in the chain than if there is only a few. This fact is important in comparing, for example, transmission in the lemniscal sensory pathways to the cerebral cortex and transmission in the reticular activating system. Since the minimum time for transmission across one synapse is half a millisecond, it is also possible to determine whether a given reflex pathway is monosynaptic or polysynaptic, so contains one synapse or more synapses, by measuring the delay in transmission from the dorsal to ventral route across the spinal cord. More than 50 chemical substances, as I mentioned already before, have been proved or postulated to function as synaptic transmitters. Actually, in the table, you can see the important characteristic of variable neurotransmitters. One group comprises small molecule rapidly acting transmitters. The other is made up of a larger number of neuropeptides of much larger molecular size, which usually act much more slowly. A few gaseous molecules such as nitric oxide, hydrogen sulfide, and carbon monoxide may also serve as transmitter modulators. The small molecule rapidly acting transmitters cause most acute responses of the nervous system, such as transmission of sensory signals to the brain and motor signals back to the muscles. The neuropeptides, in contrast, usually cause more prolonged actions, such as long-term changes in numbers of neuronal receptors, long-term opening or closure of certain ion channels, and possibly even long-term changes in numbers of synapses or sizes of synapses. And let us characterize some important small molecule transmitters. Acetylcholine is secreted by neurons in many areas of the nervous system, but specifically by the terminals of the large pyramidal cells from the motor cortex, also in several different types of neurons in the basal ganglia, the motor neurons in pathways which supply skeletal muscles also secrete acetylcholine. Also preganglionic neurons of the autonomic nervous system. Acetylcholine used for conduction excitation for postganglionic neurons of the parasympathetic nervous system. And also this neurotransmitter is acting in some of the postganglionic neurons of the sympathetic nervous system. In most cases, acetylcholine has an excitatory effect. However, it is known to have inhibitory effect and some peripheral parasympathetic nerve endings, such as inhibition of the HAT by vagus nerves. The next important neurotransmitter is norepinephrine, which is secreted by the terminals of many neurons, whose cell bodies are located in the brainstem and hypothalamus. Specifically, norepinephrine secreting neurons located in the locus coeruleus in the pons send nerve fibers to widespread areas of the brain to help control overall activity and mood or mind, such as increasing the level of wakefulness, or so on. In most of these areas, norepinephrine probably activates excitatory receptors, but in a few areas is effect of activation inhibitory receptors instead. Norepinephrine is also secreted by most postganglionic neurons of the sympathetic nervous system, where it excites some organs but inhibits others. Dopamine is secreted by neurons that originate in the substantia nigra. The termination of these neurons is mainly in the striatal region of the basal ganglia. The effect of dopamine is usually inhibition. Glycine also is important neurotransmitter, is secreted mainly at synapses in the spinal cord. It is believed to always act as inhibitory transmitter. One more important substance, gamma-aminobutyric acid, or GABA, is secreted by nerve terminals in the spinal cord, cerebellum, basal ganglia, and many areas of the cortex. It is the primary inhibitory neurotransmitter in the adult central nervous system. Yet in the early stages of brain development, including the embryonic period and first week of postnatal life, GABA is thought to serve as an excitatory neurotransmitter. The next important neurotransmitter is glutamate, which is secreted by the presynaptic terminals in many of the sensory pathways entering the central nervous system, as well as in many areas of the cerebral cortex. It probably always causes excitation. Serotonin is secreted by nuclei that originate in the median rave of the brain stem and project to many brain and spinal cord areas, especially to the dorsal horns of the spinal cord and the hypothalamus. Serotonin acts as an inhibitor of pain pathways in the cord, and inhibitor action in the higher regions of the nervous system is believed to help control the mood of the person, perhaps even to cause sleep. One more important substance is nitric oxide, which is produced by nerve terminals in areas of the brain responsible for long-term behavior and memory. Therefore, this gaseous transmitter might in the future explain some behavior and memory functions that those far have defined understanding. Nitric oxide is different from other small molecule transmitters in its mechanism of formation in the presynaptic terminal and in its actions on the postsynaptic neuron. It is not performed and stored in vesicles in the presynaptic terminal, as are other transmitters. Instead, it is synthesized almost instantly as needed, and then diffuses out the presynaptic terminals over a period of seconds rather than being released in vesicular packets, and next it diffuses into postsynaptic neurons nearby. In the postsynaptic neuron, in this neuron, it usually does not alter the membrane potential greatly, but instead changes intracellular metabolic functions that modify neuronal excitability for seconds, minutes, or perhaps even longer. Neuropeptides are synthesized differently and have actions that they are usually show, and in other ways different from those of the small molecule transmitters. The neuropeptides are not synthesized in the cytosol of the presynaptic terminals. Instead, they are synthesized as integral parts of large protein molecules by ribosomes in the neuronal cell bodies. And this is different from functioning of electrical synapses, which you actually see on the slide. Electrical synapses, in contrast, are characterized by direct open fluid channels that conduct electricity from one cell to the next. Most of these consist of small protein tubular structures called gap junctions that allow free movement of ions from the interior of the cell to the interior of the next. And only a few gap junctions have been found in the central nervous system. However, it is by way of gap junctions and other similar junctions that action potentials are transmitted from one smooth muscle fiber to the next in visceral smooth muscle, and from one cardiac muscle cell to the next in cardiac muscle.
In order to provide communication among the cells of the body, the billions of neurons in the nervous system must first communicate with one another or with effector cells via synapses. Synapses allow information to be filtered and integrated. Synapses are either electrical or chemical. Electrical synapses are called gap junctions and have many ion channels called connexons connecting two adjacent cells. Electrical synapses allow quicker communication, more coordinated communication between cells, and two-way transmission. A physical separation between adjacent cells, called the synaptic cleft, is a characteristic of chemical synapses. Because action potentials cannot cross a synaptic cleft, this electrical signal is converted into a chemical signal using neurotransmitters to cross the cleft. Unlike electrical synapses that allow for two-way transmission of signals, communication at a chemical synapse is one way only, from presynaptic to postsynaptic membranes.
The interneuronal connections may function with variable special effects, which schematically are presented in the picture. And first of all, let us characterize the effect of spatial summation. Spatial summation in neurons. This is a process which obtains a threshold for firing excitation of a single presynaptic terminal on the surface of a neuron almost never excites the neuron. So the amount of transmitter released by single terminal to cause the EPSP is usually not more than half to 1 millivolt instead of the 10 to 20 millivolt normally required to reach threshold for excitation. However, many presynaptic terminals are usually stimulated at the same time, even though these terminals are spread over wide areas of the neuron, their effects can still summate. That is, they can add to one another until neuronal excitation occurs. When the excitatory postsynaptic potential becomes great enough, the threshold for firing will be reached and an action potential will develop spontaneously in the initial segment of the axon. This effect of summing simultaneous postsynaptic potential by activation multiply terminals or widely spaced areas of the neuron membrane is called spatial summation. So impulse comes from different directions. There is also effect of temporal summation. Temporal summation caused by successive discharges of a presynaptic terminal. Each time a presynaptic terminal fires, the released transmitter substance opens the membrane channels for at most 1 or 2 milliseconds. However, the charged postsynaptic potential lasts up to 15 milliseconds after the synaptic membrane channels have already closed. Therefore, a second opening of the same channels can increase the postsynaptic potential to a still greater level. And the more rapid the rate of stimulation, the greater the postsynaptic potential becomes. Those successive discharges from a single presynaptic terminal, if they occur rapidly enough, and they then can add to one another, that is, they can summate. This type of summation is called temporal summation. Simultaneous summation of inhibitory and excitatory postsynaptic potentials is also possible. If an inhibitory postsynaptic potential tends to decrease the membrane potential to a more negative value, while excitatory postsynaptic potential tends to increase the potential of the postsynaptic membrane. These two effects can completely or partially nullify each other. Thus, if a neuron is being excited by EPSP, an inhibitory signal from another source can often reduce the postsynaptic potential to less than threshold value for excitation, thus turning off the activity of the neuron. There is also a special effect of interneuronal connection, which is characterized as facilitation of neurons. Often the summated postsynaptic potential is excitatory, but has not risen high enough to reach the threshold for firing by the postsynaptic neuron. When this situation occurs, the neuron is said to be facilitated, that is, its membrane potential is nearer the threshold for firing than normal, but is not yet at the firing level. Consequently, another excitatory signal entering the neuron from some other source can then excite the neuron very easily. Diffuse signals in the neurons in the central nervous system often do facilitate large groups of neurons so that they can respond quickly and easily to signals arriving from other sources. And there is also a specialty of functioning of dendrites for exciting neurons. Large spatial field of excitation of dendrites is revealed in histological research, and the dendrites of the anterior motor neurons often extend 500 to 1000 micrometers in all directions from the neuronal soma, and these dendrites can receive signals from a large spatial area around the motor neuron. This feature provides a vast opportunity for summation of signals from many separate presynaptic nerve endings. It is also important that between 80 to 95% of all presynaptic terminals of the anterior motor neuron terminate on dendrites, in contrast to only 5 to 20% terminating on the neuronal soma. Therefore, a large share of the excitation is provided by signals transmitted by way of the dendrites. Most dendrites cannot transmit action potentials, but they can transmit signals within the same neuron by electrotonic conductions. Actually, most dendrites fail to transmit action potential because of their membranes have relatively few voltage-gated sodium channels, and their thresholds for excitation are too high for action potentials to occur. Yet they do transmit electrotonic current down the dendrites to the soma, and transmission of electrotonic current means direct spread of electrical current by ion conduction in the fluids of the dendrites, but without the generation of true active potential. The electrical events during neuronal inhibition require special description. First of all, let us speak about the effect of inhibitory synapses on the postsynaptic membrane, which generate inhibitory postsynaptic potential. The inhibitory synapses mainly open chloride channels, allowing for easier passage of chloride ions. To understand how the inhibitory synapses inhibit the postsynaptic neuron, we must recall what we learned about the Nernst potential for chloride ions. We calculated the Nernst potential for chloride ions to be about minus 70 millivolt. And this potential is more negative than the minus 65 millivolt normally present inside the resting neuronal membrane. Therefore, opening the chloride channels will allow negatively charged chloride ions to move from the extracellular fluid to the interior, which will make the interior membrane potential more negative than the normal, approaching the minus 70 millivolt level. Opening potassium channels will allow positively charged potassium ions to move to the exterior and will also make the interior membrane potential more negative than usual. Thus, both chloride influx and potassium efflux increase the degree of intracellular negativity, and this is called hyperpolarization. The neuron is inhibited because the membrane potential is even more negative than the normal intracellular potential. Therefore, an increase in negativity beyond the normal resting membrane potential level is called an inhibitory postsynaptic potential. Presynaptic inhibition, this is actually effect which may be revealed in case when some neurons make contact with inhibitory synapse. And in addition to postsynaptic inhibition caused by inhibitory synapse operating at the neuronal membrane, presynaptic inhibition often occurs at the presynaptic terminals before the signal ever reaches the synapse. Presynaptic inhibition is caused by release of any inhibitory substance onto the outsides of the presynaptic nerve fibers before their own endings terminate on the postsynaptic neuron. In most cases, the inhibitory transmitter substance is gamma-aminobutyric acid, which opens anion channels, allowing large number of chloride ions to diffuse into the terminal fibril. The negative charges of these ions inhibit synaptic transmission because they cancel much of the excitatory effects of the positively charged sodium ions that also enter the terminal fibers when an action potential arrives. In difference to presynaptic inhibition, the postsynaptic inhibition is effect of direct contact between the inhibitory neuron to the other excitatory neuron. And postsynaptic inhibition so is caused by inhibitory synapses operating at the neuronal membrane. You can see this kind of connection in the picture. There is also a variant of lateral inhibition, when the inhibitory contact is activated for the other excitatory neuron. You can see it in the right lower corner of the picture. Virtually every sensory pathway, when excited, gives rise simultaneously to lateral inhibitory signals. These inhibitory signals spread to the sides of the excitatory signal and inhibit adjacent neurons. For example, consider an excited neuron in dorsal column nucleus. Aside from the central excitatory signal, short lateral pathways transmit inhibitory signals to the surrounding neurons. That is, these signals pass through additional interneurons that secrete an inhibitory neurotransmitter. And the importance of lateral inhibition, also called surround inhibition, is that it blocks lateral spread of the excitatory signals and therefore increases the degree of contrast in the sensory pattern perceived in the cerebral cortex. In the case of dorsal column system, lateral inhibitory signals occur at each synaptic level. For example, the dorsal column nuclei of the medulla or the ventrobasal nuclei of the thalamus or the cortex itself. At each of these levels listed before, the lateral inhibition helps to block lateral spread of excitatory signal, and as a result, the peaks of excitation stand out and much of the surrounding diffuse stimulation is blocked. Skeletal muscle fibers are innervated also by chemical synapses. And as I said you before, actually we describe the sequence of events in chemical synapse based on example of neuromuscular junction. So skeletal muscle fibers innervated by large myelinated nerve fibers that originate from the motor neurons in the anterior horns of the spinal cord. Each nerve ending makes a junction called the neuromuscular junction, with the muscle fiber near its midpoint. The action potential initiated in the muscle fiber by the nerve signal travels in both directions toward the muscle fiber ends, with the exception of about 2% of the muscle fibers. There is only one such junction per muscle fiber. The nerve fiber forms a complex of branching nerve terminals that invaginate into the surface of the muscle fiber, but lay outside the muscle fiber plasma membrane. The entire structure is called the motor end plate, which actually you can see in the picture. It is covered by one or more Schwann cells that insulated from the surrounding fluids, and in the axon terminal are many mitochondria that supply adenosine triphosphate or ATP, the energy source used for synthesis of a neurotransmitter acetylcholine, which excites the muscle fiber membrane. Acetylcholine is synthesized in the cytoplasm of the terminal, but is absorbed rapidly into many small synaptic vesicles, about 300,000 of which are normally in the terminals of a single end plate. In the synaptic space are large quantities of the enzyme acetylcholine esterase, which destroys acetylcholine a few milliseconds after it has been released from the synaptic vesicles. When an action potential spreads over the terminal, the voltage-gated calcium channels open and allow calcium ions to diffuse from the synaptic space to the interior of the nerve terminal. The calcium ions in turn are believed to activate calcium calmodulin-dependent protein kinase, which in turn phosphorylates synapsin proteins that anchor the acetylcholine vesicles to the cytoskeleton of the presynaptic terminal. This process frees the acetylcholine vesicles from the cytoskeleton and allows them to move to the active zone of the presynaptic neuronal membrane adjacent to the dense bars. The vesicles then dock at the release sites, fuse with the neural membrane, and empty the acetylcholine into the synaptic space by the process of exocytosis. Actually, in the picture, you may see many small acetylcholine receptors and voltage-gated sodium channels in the muscle fiber membrane. The acetylcholine-gated ion channels are located almost entirely near the mouth of the subneural clefts, laying immediately below the dense bar areas, where the acetylcholine is emptied into the synaptic space. The voltage-gated sodium channels also line the subneural clefts. The channel remains constricted, as actually shown in part A of the figure, until two acetylcholine molecules attach respectively to the two alpha subunit proteins. This attachment causes a conformational change that opens the channel, as shown in part B of the figure, and the acetylcholine-gated channel has a diameter of about 0.65 nanometer, which is large enough to allow the important positive ions, sodium, potassium, and calcium, to move easily through the opening. The principal effect of opening the acetylcholine-gated channels is to allow sodium ions to flow to the inside of the fiber, carrying positive charges with them. This action creates a local positive potential change inside the muscle fiber membrane called the end plate potential. The skeletal muscle fiber is so large that action potentials spreading along its surface membrane cause almost no current flow deep within the fiber. Maximum muscle contraction, however, requires the current to penetrate deeply into the muscle fiber to the vicinity of the separate myofibrils. This penetration is achieved by transmission of action potentials along transverse tubules, also called T-tubules. You can see them in the picture, that penetrate all the way through the muscle fiber. The T-tubule action potentials cause release of calcium ions inside the muscle fiber in the immediate vicinity of the myofibrils, and these calcium ions then cause contraction. The overall process is called excitation-contraction coupling. You have studied in details in the biochemistry department all this process, but you need to know this for understanding mechanism of muscular contraction in physiology course. As the action potential reaches the T-tubule, their voltage change is sensed by dihydropyridine receptors linked to calcium release channels, also called ryanodine receptor channels, in the adjacent sarcoplasmatic reticular cisterna. Activation of dihydropyridine receptors triggers the opening of the calcium release channels in the cisterna, and as well as in the attached longitudinal tubules. These channels remain open for a few milliseconds, releasing calcium ions into the sarcoplasm surrounding the myofibrils and causing actually the process of contraction.
Skeletal muscle cells contract as a result of impulses from motor neurons. The place where a motor neuron stimulates a muscle cell is called a neuromuscular junction. Your goals for learning are to examine the structure of a neuromuscular junction, to understand the sequence of events occurring at the neuromuscular junction following a stimulus. Here's what you need to know. Anatomy of a skeletal muscle cell. Definitions of resting membrane potential, action potential, and depolarization. To review the anatomy of a skeletal muscle cell, click the link button. If you use a link button, you can return to the page you started from by clicking the return button. To see definitions of terms, click the bold red words. Skeletal muscle cells are electrically insulated from each other by endomysium. In order for skeletal muscle cells to contract, each cell must be stimulated by a process of a motor neuron. Each motor neuron is a single nerve cell extending from the brain or spinal cord to the muscle, where it can stimulate several muscle cells. The axon is the elongated process of the nerve cell that carries impulses to the muscle. The neuromuscular junction is the place where the terminal portion of a motor neuron axon meets the muscle cell membrane. Click the neuromuscular junction to see it in an expanded view. Now let's take a closer look at a neuromuscular junction. The swollen distal end of the motor neuron axon is called the axon terminal. Within the axon terminal are synaptic vesicles containing the neurotransmitter acetylcholine. Notice that at the neuromuscular junction, the motor neuron and the sarcolemma of the muscle cell do not actually touch. The synaptic cleft is the space between the axon terminal and a folded region of the sarcolemma called the motor end plate, highlighted here in red. Recall that the sarcolemma is the general name for the muscle cell membrane. The T-tubules are invaginations of the sarcolemma penetrating deep into the interior of the cell. The terminal cisternae are specialized regions of the sarcoplasmic reticulum that serve as reservoirs of calcium ions, shown here as red dots. Portions of the sarcoplasmic reticulum have been removed here in order to see the underlying structures. The sarcomere is the contractile unit of the muscle cell that extends from one Z-line to the next. The plus and minus signs on the axon terminal, sarcolemma, and T-tubules indicate a polarized condition called the resting membrane potential. Although plus and minus signs are not shown on the motor end plate, it is also polarized. Here's an overview of the activity that occurs at the neuromuscular junction. An action potential arriving at the axon terminal triggers the release of the neurotransmitter acetylcholine, resulting in the depolarization of the motor end plate. This depolarization triggers an action potential that propagates along the sarcolemma and down the T tubules, causing contraction of the sarcomeres. On the next several pages, we will look at this process in more detail. When the action potential arrives at the axon terminal, the voltage change of the membrane opens voltage-regulated calcium channels, allowing calcium ions to enter the axon terminal. To see the arrival of the action potential, click the motor neuron. Click the calcium channel to see calcium ions enter the axon terminal. The calcium ions cause several synaptic vesicles to fuse with the membrane of the axon terminal. Click a synaptic vesicle to see this activity. The neurotransmitter acetylcholine contained within the vesicles is liberated by exocytosis into the synaptic cleft. Additionally, calcium ions are pumped out of the axon terminal. Click a fused synaptic vesicle to start exocytosis. Acetylcholine binds to receptor sites of chemically regulated ion channels on the motor end plate. This causes the channels to open, permitting an influx of sodium ions and a small efflux of potassium ions. This ion exchange causes a local depolarization of the motor end plate, which will be indicated here as a glow. Click the acetylcholine molecule to start this process. Now the motor end plate is depolarized. After a brief period of time, acetylcholine diffuses away from its receptor site, and the ion channel closes. Acetylcholine is then broken down by the enzyme acetylcholinesterase. Click the acetylcholinesterase molecule to start this process. The depolarization of the motor end plate initiates an action potential which propagates along the sarcolemma in all directions and down the T tubules. Click the depolarized motor end plate to initiate the action potential. The action potential causes the release of calcium ions from the terminal cisternae into the cytosol. Click the terminal cisternae to see this activity. Calcium ions trigger a contraction of the muscle cell. This process will be examined in more detail in sliding filament theory. Click the muscle cell to see it contract. To see the activity at the neuromuscular junction as a continuous process, click the motor neuron. Here's a summary of what we've covered. Each skeletal muscle cell is individually stimulated by a motor neuron. The neuromuscular junction is the place where the terminal portion of a motor neuron axon meets a muscle cell membrane, separated by a synaptic cleft. An action potential arriving at the axon terminal brings about the release of acetylcholine, which leads to depolarization of the motor end plate. Depolarization of the motor end plate triggers an action potential that propagates along the sarcolemma and down the T tubules. This action potential causes the release of calcium ions from the terminal cisternae into the cytosol, triggering contraction of the muscle cell. To test your knowledge, click the quiz button to go to the self-quiz.